I think the last 10 years on dragonfly. Mostly, I focused on the larval stage and the exuviae stage. Why this stage? Because simply there was only little knowledge about this topic. Well, actually, it's the most important, not the most important, but a very long time when in their lifespan that they have as a larvae. So this is why I try to focus on this part of their life, and then once you start discovering this world, it's actually, you will realize it's really fascinating. There are a lot of books on dragonflies around the world, but the one who does speak about larval stage, they are much more rare. The oldest one I know, maybe they are older, but I just don't know them, is from a Dutch author, Jan Swammerdam, in 1669, in his book about all kind of living things. He spoke about the several stages of the dragonflies, and he has actually, it's in Dutch. He called them wormpopke. And wormpopke, if I translate it to English, it will be like a worm cocoon or something like that. This was the name that he gave for the larvae and the exuviae. Of course, later other books were published talking about these exuviae and sometimes of the larvae. Like, for example, one of the major book in Europe was at the time was the book of SQ in 1988. Philippe Courbet, in his huge book about dragonfly behavior, speak a lot also about larvae, but the major part of his book is about the adult stage. But there's still very interesting thing in his presentation of the larval stage and the exuviae as well. For Czech Republic, I've made some. I don't have all the books of there. One of the major book I have, it's about the adult mostly. This huge book of the Atlas of Czech Republic, and there are several books also published describing, for example, like this one, the adult, and also having chapters on how to identify exuviae, for example here. And of course, a book from Martin Waldhauser and Martin Cerny as well. This book, I like it a lot because it has also a lot of photographs of the larval stage, not only the exuviae. And well, I think for almost all the species in the book have not only the adult but also the larval pictures. Book that only speaks about larvae and not, for example, like the adult, there are not that much. Gianmaria Carcini, in 1988, he is still working on, by the way, on books on dragonfly larvae. Wrote one in Italian with wonderful drawing. And Gerke Stenberg, I really like that book, especially at the time for exuviae work. It was one of the major book, and they was one of the first to have attempt having a European key inside the book, and what they really realized that there was a lot of knowledge lacking. And what I do like in this book, for example, is once you have very rare species like Aeshna crenata from Scandinavia, they say also, for example, building the key, how many exuviae they use, because sometimes it was only building a key with 1 or 2 or 3 exuviae, which is not that much. But it was at the time the only exuviae known for that species. Later on, in 2002, came the book of Heidemann und Seidenbusch. This one was definitely one of the, well, for many years, one of the book that everybody used because it has all the knowledge from the time. Some other book, for example, like England, like from Steve Cam from the British Isles. It is really useful there, but of course for Europe it was not complete. There is an author I would like to speak about, and it's Mr. Paul-André Robert. He died in 1977, and this was, I think, an amazing person. And why I do speak about this person is because he was one of the person who really dedicated his life to the work of dragonfly larvae. He was author of books, for example, for the text, but also he was really a keen painter. Here, this is the guy in the field. And well, he comes from a whole family having painters. His father and grandfather were making portraits for the living, but his father also focused illustrating books like butterflies or flora from the Alps. He comes from Switzerland, so he's French-speaking, but also, of course, he has for the whole Switzerland, also the German-speaking part, also a huge impact. At the time, his books were about insects, and he wrote a book about dragonfly, Les Libellules, Die Libellen in German, in 1958, translated a year later in German. And that book became for me really one of the major books, especially when it becomes to dragonfly study, because it had really a lot of biology information about the adult, but also about the larvae. And when I was writing a book about the larvae of dragonfly, Hans Rudi Wildermuth, was professor in Switzerland, told me, "Do you have the book of Robert?" And I said, "Of course I have it." And he said, "No, not the adult one, the larvae one." And I never heard about this larvae book of Robert. And then we realized the manuscript was finished, but the book was never published. So we went to Switzerland and searched for this, let's say, masterpiece of larval book on dragonfly. And in a museum, we found this manuscript. And actually, Robert died, as I said, in 1977, but the book was finished. And this is Hans Rudi looking with a loop to Calopteryx painting. And when we saw those drawings, they were simply amazing. The book was, all the plate was rather well preserved. This one, by the way, one you see, is one of the most damaged, with all those, let's say, old dots that comes when it's when the paper gets old. But the major problem was that painting here should have come here in this line drawing space, because he made some inked line drawing, and the aquarelle, like this one, should have come here. Also the lamellae should have come as well here, for example. And at the time, it was like a huge amount of work to do, because they didn't have Photoshop or something like that. This is Robert at the age of 14, 16, and this is already, for example, the drawing he made at the time for Codulegaster boltoni. And when I realized actually this drawing were like almost perfect. For example, this is a drawing of Leucorrhenia caudalis he made, like, I don't know exactly, maybe something like 1960. And 50 years later, I make a picture, for example, and then you realize it's impossible that it's the same individual, of course, but then you realize how perfect was the drawing. Also, for example, if you look to this photograph of Pyrosoma nymphola, and this is the detail of the lamellae, and this is the drawing Robert made. This is, he respects well the color, but it was even better, which is very, very important in Odonata study when they are as a larval stage. This is a line drawing, for example, of a Primentum of Eschnasianea. This is a picture I made of the same species, and the most important, especially when it comes to measurement and Primentums to identify the species, are the proportion. And if you look, for example, here, this is the combination of both. Well, it's perfect, really perfect. And what we have done with a team of almost 15 people, we have made this book finally to a fully book. The book is, I guess, sold out now. It was also a rather expensive one, but at least it was on paper finally, because Robert spent almost 50 years working on that one. And now that the book is finished and actually sold out, I'm talking to the director of museum and also to the Dutch editor. to have this book in a free open source as a PDF. I'm working on that, and I will definitely let you know once it's finished, because it covers not all the species. For example, species that Tritemis, for example, there are only a few, but also other species that were not discovered. And but still, it's really a good reference to have it. It's written in French and in German. With Evert for the Ploch, I work also for several years on Northwest Europe. This was actually one of the try we made in Dutch to have the knowledge about the dragonfly larvae in one book. And I consider this book as a good exercise for the book I'm still working on European dragonflies. There will be at the end, I don't know when, maybe 1 year or 2 years of work. There will be at the end, I guess, 2 books. One that specializes of exuviae of European dragonflies with a key inside, and one that you can see as a complementary book to that exuviae book will be about the larvae and their biology. Here, for example, that book of larvae, it has a lot of attention to anatomy, because it's really important to, especially when you work with key or description, to have the proper name. And when you look to the book, you have, I liked still to have the match with the adult, for example, and we add also a biotop pictures, and then like a general pictures of the species as larval stage, and then some details that are important. And this is a reference to the other book I wrote 2 years before that one, that refers to the key part of the exuviae chapter, and also the description of the exuviae species. This is, for example, for Zygoptera, and this is for Anisoptera. This is how the book looks like. These books are not sold out, but definitely one day, once they are finished and sold out, I'm really attempting to, once the publisher say we will never publish it again or make a reprint or something like that, then I will do my best to have all those books as open source and free to everyone. Well, if you look, for example, to Ashna viridis, which is a very rare species in Holland, but it's a species it's rare species, but it's rather common at some places where close to where I live, and I use it for that one to start actually the biology of the larvae. Well, when do you start actually in a life cycle? Well, I decided to use the copula, then the female lay eggs, and then after that you get a larvae, and this larvae will emerge to an adult. Well, first of all, actually in Europe, we don't use the proper name. It isn't a larvae, it should be a nymph, but somehow the name larvae stayed in the, I will say, as a common name instead of nymph. And why is it not a larvae? For example, a caterpillar is a larvae. A butterfly has an egg. It has also a caterpillar, thus a larvae, and then there is an extra stage, which is actually the cocoon, and then you get the adult. Well, dragonfly don't have this cocoon stage, and they go from the larvae to the adults. They skip that cocoon part, and this is why they are not larvae but nymph. But all right, when, as I said, in Northern America, for example, they are really laughing when you say, what? This is something you learn at school. It is not a larva, it's a nymph, but somehow, even when you put in titles, for example, of books, it has, for Europe, it has always been wrongly used. But okay, for the common name, I prefer to use that one as well. So this is why I keep on larvae. The adult stage of many dragonflies, it's sometimes some weeks, sometimes some months. Even, for example, like in those winter damselfly, for example, from the Lestidae, like Sempecma, they stay really a long time as an adult. But most of the time, it's in months. While actually for some species of dragonfly, the larvae can stay up to 5 years underwater. And when a female lay eggs, it really depends. For example, Chalcolestes viridis, well, it will lay eggs on branches out of the water, and some species put eggs in dry sand, and some species put eggs, and like you see in Eschnaviridis, on Stratiotes, this water soldier plant, and they use actually they go sometimes really deep in the water and they try to put their eggs in the submerged part of the plant. But sometimes dragonflies have a strange habit. For example, Boyeria irene or Boyeria cretensis, those two eschnids, well, they lay eggs in the most darkest part of the river. And I have a friend who was with his girlfriend close to the river in the Ardèche from France, and suddenly the dragonfly female flew under the skirt of his girlfriend. Well, you can imagine you have this lady dancing in the river because it's not she was not very pleased of that. But what did happen actually is this is the biotop that Boyeria cretensis, this endemic to the island of Crete, lay eggs. This part, this is the place she will try to search. And it was rather funny because when you see this female, when I was walking in the water searching for the larvae and try to understand their biotopes, and they're searching also for the exuviae, when I realized this was not the darkest place in the river, the darkest place was actually, in fact, my shoes. So this very rare dragonfly directly circle around me, show these black shoes, and try to lay egg on it. So I called Eva, the friend I was on the field with that day, and I say, Eva, come and have a look, because it's really rather amazing to see this female trying to lay egg on this spot. And when he arrived, the female decided that my shoe was definitely not the proper way to have this to lay eggs. So she tried directly to the eggs, putting eggs in the shoes of Eva. Well, of course, you don't see all those strange behavior, but this is rather interesting to see that at the moment the female lay eggs, they do that like on a very careful way. Where here they show the darkest place, and when they see it's not suitable, and after a few seconds they flew away. But for example, in Libellula depressa, you can have sometimes at some places they just tip the water with their abdomen, and you can have like a huge amount of tiny eggs. They are like a few millimeters, and after several times, it can go from 1 to a week. Lestes drias, for example, has a very short egg and larval stage. Within several weeks, they can fly away. And this is actually what you call the proto-larvae, just coming out of the eggs. And for example, when you have birds, most of the time within like a girl, herring girl, for example, well, the bird, of course, grow rather quickly. For example, if you look to a swan, it's amazing that a swan living in a tundra within, I don't know, a few weeks comes out of an egg, reach the length of 1.60 meters, and is able to travel a migration of almost 20,000 kilometers. And but here in dragonflies, if you look in the water, you will see actually that even if you have a juvenile one, for example, you will have several stages. And why? Simply because they molt on the water. This is, for example, Codylegaster bidentata, but this is also Codylegaster bidentata, and this is a fully grown. This one will emerge the season later, but this one may take several years before they grow completely to the size that you call it F0, to the size that they will actually emerge. It's not known yet about this transition, how many molting. Most of the species molt about 10 to 15 times underwater. And but the thing is, sometimes you see, for example, like a big larvae, and especially in Codylegaster, because they really can stay several years underwater. But it doesn't mean that there is, for example, a lot of food. Most of the time, well, this is not fully tested yet, but they are more hypothetical, that in fact, when you have larger larvae of Codylegaster, it's because they don't have enough food underwater to get, let's say, the amount of energy they need to emerge quickly, so they stay underwater and they molt and get bigger, and at one point they have enough energy and they will emerge. This is our Echinacea, for example, molt underwater. Here you have this black old skin, let's say, like this, and you will have this fresh new green. Remember this greenish color, because I will talk about this later. It's really amazing. And at the end, this is a moment, by the way, if you look on dragonfly larvae, be careful once they are in this type of color, because they are very fragile. Within dragonflies in Europe, there are two large groups of dragonflies, and I don't know the knowledge you have on this topic, so I just explained the adult a bit, but I will definitely focus on the larvae part. This is, for example, Somatochloria lunulatum. It's a Zygoptera. And this is Epithecla bimaculata, an Anisoptera. And most of the Zygoptera, they have their wings along the body, while the wings of Anisoptera are more spread. Lestidae, for example, are called in English spread wings because they are Zygoptera, a kind of exception, but they can spread their wings more. But in fact, in Zygoptera groups, let's say all the species except one that I will also discuss about, they have, as a larval stage, they have 3 lamellae, caudal gills, you can call them at them. They use it, for example, for swim, but also for breathing. While the Anisoptera have different shape, they don't have any caudal gills, never, but they do have, for example, dorsal spines, lateral spines, even in Epithecla, spines on the head, and they breathe. Here the water is coming in and out, and the branchies are here. By the way, Anisoptera, Zygoptera, sorry, they also sometimes, I will discuss also about this, they can lose them, but they still have the possibility to breathe. What I will do now actually is present you all the, not only the, let's say, the family, but to go sometimes into details of this, of all the families from Europe, and describe them shortly as an adult, but also as the larvae. This is, for example, Calopteryx splendens, and this group of damselfly are, as a larvae, well, as an adult, they are very recognizable. The male are mostly greenish and bluish, and with the blue on the wings, while the female is greenish. And the larvae are also really easy to recognize because they are very long lamellae, but if you look on the head, the first segment of the antennae is very, very long. In fact, the first segment is longer than all the segment others together. So this makes it actually within the field, you can rather easily recognize this species. Another family, the Lestidae, this is Lestesponsa, and Lestesponsa is typical a species that can be very, very common at some moment. And as a larvae, well, you just have to search in place with a lot of sphagnum mosses, for example, and then you will find some larvae. This is this Lestesponsa, for example. This is interesting because you see this 3 gill. Actually, you see 2, but the other one is in the back here. There are 2 paraprocts of paragills and 1 epiproct. It is this part here. And you can see the prementum, the eyes, the abdomen, but also, for example, this one, it's the wings are not stick to the body, but you can see some kind of structure inside these wings. Well, actually, this is the place where the wings will be when it emerge. And when you see that, then you can say, oh, it will emerge soon. Most of the time, when they don't have that, the wings are close to the body. This is, for example, Lestes macrostigma, and while Lestesponsa had a lot of bands on the gills, Lestes macrostigma doesn't have only a few bands, and sometimes, for example, these typical white tips. And here you see, for example, the wings are clear and stick to the body. You have another family, which is, well, the biggest one in the Dancer Fly group, the Cenagrionidae. This is Sonagrion armatum, a rather rare species because it's a Nordic species. In Holland, there is only one spot. For example, you have to go to northern Germany or Denmark and even to Scandinavia to see that one. But their larvae is rather typical as a Sonagrionid larvae. It has, look, you can see clearly here, this is definitely not Calopteryx because it doesn't have that long first segment of antennae. The first segment is here, so it's very small. But you can see here the caudal gills, they have separation here, which is the nodal line. And not all the species have that, but this one here has. And even the group of several species has also a thicker border here. This is a group of Armatum, Sonagrion lunulatum, and Sonagrion astulatum. And this, why do they have that? Simply, I don't know. But it's useful to separate those species. In Europe, there is a strange Sonagrionid, this one, Sonagrion Hylas frey, which is actually, it's called in English, I guess, the Siberian Dancer Fly. And actually, it's you can find it in Austria in about 8 or 9 lakes or something like that. And that's it. It was also a long time ago in Switzerland, but it disappeared there. But this species is kind of a relict species from a glacial time. And you can find the Hylas, that species in Siberia. But the strange thing, it is definitely a Sonagrionid, but the caudal gills, they don't have this nodal line. And some of the species, like for example, Sonagrion ornatum, like this is one adult of it, well, the caudal gills, the nodal line is up to here, so it's very far away. So there is a huge variation with the morphology of those gills, which is very useful. And even, for example, once you have to identify species, most of people say, oh, we'll stick on the Anisoptera because they are bigger or something like that. But most of the time, the Zygoptera are even more easy. Not all of them, of course, there are always exceptions, but most of them. Here, the last group of the common species of Zygoptera, Platycnemidae. This is Platycnemis pennipes, and this group, for example, is one of the group I just don't know. For the moment, they are in Europe. There are 3 of them: Platycnemis pennipes, Latipes, and Acutipenis. If you have trouble sleeping, contact me. I will give you some work to do on the key to these 3 species because I spend a lot of time and I really don't know. So for the moment, it's unknown as a larval stage to separate those species. There is, for example, another one, which is Acutipenis, completely different as an adult. There is no way this is not pennipes. But well, for example, these gills here are completely different. So you could say, oh, maybe it's the form of them. But I have tried. The problem is mostly Platycnemis pennipes, the common one. The gills of pennipes can vary from, like this one as well, like Acutipenis, but also very broad and very slender. So there is too much variation to use it as a useful criteria to separate species. And the last, let's say, family with one species in Europe of Zygoptera, and I really have to say, if one day you have the possibility to travel east, go there because it's wonderful. This is the female, for example, of the Epalac fatim. And why do I speak of this species? It occurs also, for example, in Bulgaria, for example, you can find already this species. And maybe one day in Czech Republic would be nice, but it's possible. And but that species is, for example, it's amazing because I told you that the larvae of dancer fly, they have 3 gills, which are actually flat, and they use them for swimming. But this one here is completely different. It has, at the end of the abdomen, it has no 3 gills, it has 3 balloons. And this balloon, actually, they are very strange. The behavior, by the way, of the larvae is also very strange because it doesn't swim around. It's more like a Plecoptera. It will stay on stone. You can see very strong legs because it likes streaming water. But also, the very strange, one thing we know for sure is that this, well, it's a gill for the other Zygoptera, these are not the gill. They don't use it for breathing, because when you look from the side, you still see the balloon here of Epalacphatim, but these are the gills. The gills are on the abdomen. So but what is the function of this? Well, this is the proof that there is a lot of research to do on that. Nobody knows. Here's a picture of one looking at me. Very strange, very strange larvae. This was the group of the Zygoptera. Now I go to the group of Anisoptera, and within you have the first group is the Ashnids. Well, I have to say, well, the adults are really beautiful to observe. The larvae are also, of course, beautiful, but they don't have much variation. For example, this is, for example, Ashna affinis, and they don't have dorsal spine. They do have lateral spines. Sometimes some species does have like a relict dorsal spine on segment 9. By the way, all the larvae, whether Zygoptera or Anisoptera, they all have 10 segments. And it's really difficult to count them from, should be somewhere like here, 1, 2, 3, and so on. The best is to start from the tail, which is this here. And for the Anisoptera, you have 1 epiproct, 2 paraprocs, and these are 1 cerci, and the tiny dot in the unsharp part here is the other cerci. And this, once you know that this is the end, this is then 10, 9, 8, and so on. And this Anisoptera from the Ashnid groups, well, one of the biggest difference you can see between all of them are the shape of the eyes. For example, this is the adult of Brachytron pratense. This is an Ashnid who has actually the smallest eyes for this group. You have a big larvae with very small eyes. This is the adult of Anax parthenope, and Anax parthenope has this kind of, I would say, almost egg-shaping eyes. Or, for example, freshly emerged Eschna subartica female, and most of, well, actually not most, but all the Eschna, they have much rounder eyes, but you see that the shape of the abdomen is rather the same. So, in fact, when I was working on Exuvier, building a key to separate species from Northwest Europe, I spent like a week and a half working on all the Zygoptera, and I spent more than 2 or 3 months working only on the Ashnids, because they are big, but they are really not that easy to separate. You really have to make measurement, especially, for example, the biggest one, actually, the like Anax. The larvae, it's almost like the biggest the larvae, the more difficult they are to separate. Another family, completely different now. These are the gomphids, and this is Onychogonphus. This is Onychogonphus falcipatus, and Onychogonphus, it's a group of gomphids, so they are definitely Anisoptera, no caudal gills, as like Zygoptera. They have flat antennae, and some of them, like Onychogonphus, they do have dorsal. It's not like it's a spine, it's more or less like it's a lobe or something like that, and they do have lateral spines. This is another genus, Paragonphus genei, Mediterranean, actually an African species. You can find a lot of species of Paragonphus in Africa. Yeah, the adult, you can easily recognize it with the well, yes, here is it, with this really kind of tail, typical from with Onychogonphus as it as well, but it's really different build. And also the green thorax here of Eugonphus cecilia as well as it, but this is typical different. The larvae of Paragonphus has no dorsal lobes, only minute. It's not really spine, and they have this typical antennae. You can see that on that, but the antennae basically it goes like this, and then at the point it goes from that way. You can see it on this part here. These are the, well, this species is really rare, and you have to go to Sardinia, for example, or Corsica. I think there are no records anymore for Corsica, but Sardinia is one of the last places you can see it, and Portugal then. This is completely what is actually the group within the gomphid that has the most species, the genus. Well, this is it should have been Gomphus, but now I think it's Stylurus, which I definitely agree, by the way. Stylurus flavipes, it's a typical large river species. In Holland, it has been really interesting because that species disappeared due to major pollution of the large rivers. And well, when a dragonfly disappears, it's not because the dragonfly cannot fly there, it's because the larvae cannot live underwater. And due to really high pollution, it was almost mostly in 1960 and 70, it was so polluted in the large river like the Rhine or something like that, that species could not live anymore. They were maybe they cannot bear this pollution, or also the food they eat cannot live there due to this major pollution. And now, due to better water quality management, well, this species is really getting common at some point. So it's really like there are very well bioindicators to use to see quality in and change in aquatic systems. This is the larvae of Stylurus flavipes. Why is it not Gonfus flavipes? Simply because actually the behavior is completely different, first of all, of the adult, but this segment here is really like all the gonfids you will see. Actually, if you have to make a key, whether it's a gonfid, you can say, is it a pancake or not? Well, this is not really a pancake, for example. But if you look, for example, to this gonfid here, this is a very rare one, Gonfid schneideri from Turkey. Well, if you look to the shape of this is a very young one, by the way. But this is because the wings are not developed. The fully grown larvae will go up to here, and then we will emerge. But you see, they are completely different in shape and very flattened, for example. There is even better, like the real pancake, I will say, and it's a wonderful species, Lindenia tetraphylla. I really love that gonfid, really big, and they can fly for hours, and you can, well, you really have to exercise running if you want to chase them because they are fast. And their larvae are almost like unique. You almost don't need a key actually to separate them. And this is it. Like also here, like a big pancake, you see that this one is about to emerge. Let's say this summer, the same summer, because it has rather long wings. And the larvae are of that species are very strange. They are the only one that has two. It's difficult to see on this picture, but here, for example, this is the tarse, and you have one long part here. You have the claw here, and you can see this yellow-orange part. You can see that there is a separation. They have two parts. So they have the tarse as two parts, while all the other gonfids have three. So this is a way to actually, even if you don't know the shape of Lindenia tetraphylla, which is rather easy, but you can still rely on morphological criteria, such as does it has two parts or three parts on the tarsus. Another group, and in Czech Republic, you have wonderful species of this one, like Coduligaster eros, and this is Boltoni, for example. Well, Coduligaster, it's like Platycnemis for the Zygoptera. They are really to separate them. I just don't know. Really, I don't know. This is Boltoni, for example. Boltoni, they don't have any dorsal spines. They do have lateral spines. For example, Boltoni has small one, but this is Coduligaster helladica from Greece, and they don't have, for example. This is Eros pelionensis, and this is a subspecies of Eros from southern Greece. And they do have, if you look carefully here, they do have small spines, but some, like Coduligaster picta, for example, they have tiny spine, but sometimes they have not. So people were saying that in Coduligaster, the one with spine are, let's say, the group of Boltoni, having Eros inside, for example, and Trinacria, and some dragonfly larvae without spine of Coduligaster, like Bidentata. They don't have spine, so they belong to that group. And Helladica is one of them, for example. But for example, in Picta, which occur in Bulgaria and in Turkey, for example, well, those species, there's a variation, so you cannot actually use it to separate those groups. But for example, one like Trinacria here from southern Italy, Well, this one, for example, if you have to shave, use that species because those spines are really, they are clear. So there is no problem. It does belong to that group of Boltoni. Another group of Anisoptera, these are the cordulids. And this is, well, a large cordulid, Epithecta bimaculata. I show that one to separate Zygoptera and Anisoptera. Well, it has everything everywhere. Dorsal spine, lateral spine, spine on the head. So it's a really, well, and it's a large larvae. Most of the cordulids are easily recognized because they have really long legs. And you can see not, you don't can see that in the picture here, but they have also on the cleft and the base of the perimentum, they have a kind of small cleft that none others, especially libellulid, which is the other group, they don't have it. But there's a lot of variation. For example, if I go back on Epithecta, you see those spines here, they do have a function. Well, Epithecta is well known to be found in places with a lot of fish, for example. And you can imagine when you are a fish and you have to eat something with a lot of spines, it's not, it might be tasty, but it's not like it's a pleasure to eat it. And so they developed that. You can clearly have a relation which is interesting to study. For example, in zooplankton, you have those water flea, and there is one water flea called Daphnia cuculata, and that water flea also developed very long helmet, depending whether it's in a fish system or not fish system. For example, here you have another cordulid, Somatochloria arctica, and Somatochloria arctica, it lives actually in sphagnum bogs. Well, there is no fish at all in these places because sometimes they completely dry out, but the larvae somehow manage to stay in this wet moss and survive. But they have, they don't have any dorsal spines and don't have any lateral spines. There is another one which is here. You can see it, by the way. Here there's no spines. Well, they have a lot of hairs, but there are no spine in it. And this very rare one, it's this one, Somatochloria Salbergy. Actually, I will say one of the most difficult dragonfly to find in the world, because you have to go north, very north, to the, actually, Alaska, Canada, Siberia, Northern Europe, in Scandinavia, you will find it. But you need to be lucky, because I have friends that spend weeks of work trying to find one adult. I was there one day, so I was really lucky. And but I was also very lucky to find one larvae of this, and it's just in between. They don't have any dorsal spine, but they do have lateral spines. Another group, which was actually from the group of the Cordulidae, but it's not separated completely, which is a good thing because they are completely different. It's this one. And this one, they are like amazing. This is not my picture. It's from Albano Soares, a friend from Portugal, because I've seen the adult, but I was never able to find one, to catch one to photograph, because they are flying all the time. But the larvae of this very large dragonfly, well, in the key, as I say, for the golden fig, is it a pancake or not? Well, I will say here, is it a spaceship or not? I will more or less say like this. Because when you look for them, this is, for example, two of them in leaf. This is typical of species that can live in sometimes in places with rock, but there is also, sometimes when you have streaming water, you have those typical place on the water which has not influence with a lot of current, for example, and with branch and detritus from root parts. For example, this is where they live. And when you look to the larvae completely, well, they are really amazing. So this is really one of the most, I will say, beautiful and strange larvae of Europe. And now I come to the last family, and actually the largest one of the Anisoptera, the Libellulidae, and they are really completely different shape, size, everything. This is, for example, a rare one from the south, Zygoptera, which is typical also in actually a more tropical species. But Zygoptera occurs also in, for example, like in Spain. And this one is clearly built to live in streaming water. You remember maybe the shape of this strange larvae with the balloon, Epalacphatim, look, it's actually rather the same, actually. But of course, without the balloon, it's completely different. But the habitus is about the same, and they occur also. So I don't know if they occur, but they could occur together. For example, when they have, because this one is more or less in the west. But if you look, very strong legs, and they do have lateral spines and dorsal lobes as well. Another one, for example, that also have that, but completely different in shape, is Brachytemis impartita. And this one, for example, that I will still have trouble watching this picture because I spent like a night, because it was always moving, swimming and everything. The Brachytemis, they are not like this. Calopteryx, for example, are very peaceful larvae. They don't like to swim and they just stay on their branch or something like that. That one was always, well, let's say, underwater on the hike, I would say. Here, a very common species in Europe, Liberula Quadrimaculata. These are very hairy larvae. You can see it here. They do have lateral spines and dorsal spines. And also here, maybe to respond to fish, but ones who has, for example, like Epithecla bimaculata, this strong response to fish, is a Sempetrum depreciusculum. And this one, for example, when you have carper, when they have a lot of these fish, for example, for consumption or only for fishing as a hobby, you can find this one, because sometimes they during the winter it need, let's say, an inverted summer cycle, I will say. In the summer it is dry most of the time, and in winter it's wet. But in fish ponds, sometimes in the winter they put the whole system dry, and in the summer they want the water. And what happens in, for example, you have that in Holland with the reeds, to make reeds on the roofs of houses. And in the summer they want it to have wet, to have the reeds phragmites, to have it growing. And in the winter they want to take these phragmites and they put the system dry. And this species here, the larvae does not the same as the other, because they lay eggs really late, like in September or October, and they will stay as an egg the whole winter. So they can actually, even if it's dry, this egg is like a time capsule. They will survive. And once the water is coming back, like in April or in May, they will start having their proto-larvae and grow up to such a larvae. They are small, but if you look, for example, in proportion, those lateral spines and dorsal spines are really big. On the other end, for example, you have some in between, like this one, a very rare one for Europe, Sempetrum cinnaeticum. You can find that one in Spain, for example. And it's in between. It has no dorsal spine, but it does have lateral spine. And I have to say, it's very easy when you want to put a key, for example, because there is one, as an adult, they look completely different, but as a larvae, maybe pretty much the same as Sempetrum cinnaeticum, with a small lateral spine, but they do have dorsal spines. So this is how you can recognize, like this one, Tritemis kirby. Tritemis kirby, by the way, this larvae is it can sustain all type of biotop, and it definitely are typical species from warm systems, and it's very strange. But maybe it's already found in France, for example. But like 15 years ago, if you use books like the book of Kelly Dijkstra, for example, you will see on the map it was not in Spain, for example. And now, especially in southern Spain, it become, I would say, the most common species of all. So these larvae, once they find their the suitable biotop, they can survive inside that, and then the adult can lay like a huge amount of eggs, especially for Tritemis kirby. If you are, for example, in a city with some artificial bassin, for example, and even if it's a small one, I know places that you can find 2,000 or 3,000 exuviae of Tritemis there. Another red one, Trochotemis eritrea. Well, Trochotemis, I put it here because this is typical one species, also with lateral spines and with no dorsal spines as well. So these are all the groups of Odonata. those Zygoptera and those Anisoptera. And it was for me very interesting when I was searching for the exuviae to also focus on the larvae. And sometimes you see, for example, very strange behavior, and that was when you want to photograph those larvae. And for example, I was in southern Spain, southern Italy, in Sardinia, to search for Somatochloria cerulescens, and small dancer fly. I was able to find it, and I put it in the water, and it was dead. And well, it's not like it's very photogenic or something like that. And I said, my goodness, what went wrong here? For example, the water quality is not the same, the temperature is not okay. But actually, after, so I told Eva what I was with, and I say, look, it's dead, but I don't know why. And I was not pleased, because I like to study, for example, exuviae, because once you have an exuviae, you have an empty shell, so you don't kill anything. And this is why I prefer to work on exuviae rather than on larvae to build keys. And but after a few minutes, actually, it was not dead at all. It was actually a kind of protection they use, and you can call it thanatosis. Thanatos was one of the god of death, and this is actually a protection that several species does. This is also Brachytron pratense, and normally in the water they are like this, with the leg and searching for food. But when you disturb them, they can look like almost like a branch. Or, for example, a cordulid species, Oxygastra curtisi. Well, I say cordulid, but this species will be separated. It will be a new family. But for the moment, we don't know which family. It will be a completely new one. But Oxygastra curtisi this is a typical habitus of the larvae, but once you disturb them, they can stay like this for several minutes. Another thing which is very interesting, and I don't know why they do have that, or the significance of that, is when you look to a larvae. This is a very rare one, Pyrosoma elizabethae, endemic to southern Greece. The eyes are completely different from species to another. This one, for example, has those typical bands, for example, or Sonagrion armatum with well, actually almost nothing, of a very strange achnid from East Europe, Caliashna microstigma, with these amazing eyes, actually, or very glossy, like the one of Eschna subartica, for example, that one here, or the typical shape of Cordulegaster with two eyes. Well, I will say almost coming out of the body like this. Remember this, because this is typical for some. I will show you something about that. Oh, really small and tiny eyes like Epitheka, and some of them have like a kind of color on the prementum, like this band and the small eyes. And this yellow, for example, these are all things that you can use. If you see this yellow on the prementum, most of the time you will know for sure you will have within the Libelulidae, you will have one of the group of Libellula, for example. Sometimes you see criteria with, for example, this one is a tiny one, Nehalenia speciosa. It has, for example, this mark. I will show you more pictures about this strange drawing they had on the head. Here, I would like to show you this. This is strange, actually. You see one gill, and but this one is very tiny, and there is no other one. So there is something happened here. And if you look, for example, to Pyrosoma nymphula, this is also, you have one complete ear, a smaller one here, and another smaller one here. This is very strange if you look in details. But other species has that as well, like Enalagma sciaticerum. This is, for example, the gill as it should be, but those two are actually, well, they are regenerating. And this is something very interesting. The dragonfly larvae has the ability during the molting, so the moment they change skin underwater, they can regrowth some organs. Of course, you can imagine if you chop the head of a poor larvae, it will not regrowth, it will be dead. But if you somehow a fish or something like that eat the gills, but they can regrowth. And this is very interesting because a lot of studies are made actually to understand how they have the mechanism behind that. And what I will say, it's almost science fiction, but it's already that still people are trying to understand that for also human medicine. I will say that if you have the possibility one day to have your, accidentally you have a finger, you lose a finger or arm or a leg or something like that, which is not a vital organ, I will say, then it will be wonderful, of course, to have the possibility to say regrow and have like all these kind of organs, like a new one. And it's not really like it's a science fiction, because, for example, here, when it goes to the valve of the breathing system of the larva, they now use the same type of construction also to make actually a new system of heart. I don't know exactly how it works, but it's just to give you an impression that actually the physiology and the morphology of larvae are really studied, because thanks to that they can maybe one day make new hearts for humans, for example. When I go back, for example, to the Nealenia, and I told you about this band, for example, larvae of species within the same species have a lot of variation. And if you look, for example, to a bird, of course, you will have like a herring girl. You will have a juvenile, which is brown, and the adult with this yellow beak and red, and also this gray color on the back. But for birds, the color is a very useful tool, but not for larvae, because, for example, this is Leucorrhenia caudalis, and Leucorrhenia caudalis, this one. If you remember, it was the one I showed, for example, for the book of Robert to compare his drawing. But this band, for example, okay, it's unsharp here because it goes back in the deeper field. But you see they are brownish. But this is, for example, also Leucorrhenia caudalis. So it's a huge variation in color. But there are species, they are all unique. For example, this is Aeshna grandis. Very dark one, and typical this light band here. Okay, but this is also Aeshna grandis, and this is also Aeshna grandis. So there is a kind of huge variation within the color of the same species. This is why I prefer to use spines or something that sometimes, and I have to say, even in Aeshna grandis, I use this kind of, for example, as a criteria, these 3 dots on the head. These are the place where this is the compound eye, and these are the ocelli. And these 3 dots here, I use them as a criteria. But the color is a thing I don't like to use, because there is a huge variation. And at the beginning of my presentation, I told you to remember when this larvae was molting on the water, this greenish color. In fact, something happened here which is really amazing, and there is not a lot of study has been made on this research. There should be about how it works, but this greenish color here, what does happen? This is the color of the larvae at one moment, but they will change color. For example, here, this is Chalcolestes viridis, and this is Chalcolestes viridis. The same one, when you change during their molting, when you change the environment, and this environment, the eyes look about the color of the system, and it will take some time, but they will try to change and adapt their color. Because look, this is also Chalcolestes viridis on the same plant, but now molting further. This is the moment that they will be fully grown and as a larvae and ready to emerge. But it's now yellow. So what happened? They spend a lot of energy, because they wanted to have the possibility to, 2 things, I will say. Avoid to be eaten by predators, but also to come closer to a prey. Some other species have another strategy. They say, I will definitely not change color, take me too much time and something like that. This is a typical picture, what I will show you, of Codulegaster, in this case, bidentata. I will wait maybe for a few seconds. Try to see the Codulegaster here. It's not easy, but this is the way Codulegaster is living. Actually, they bury themselves, And I told you to remember the eyes of Codulegaster boltoni that almost come out of the body. In fact, here is one eye, here is the other one, and here you have the two antennae. And I can tell you for sure the streaming water of the river is going that direction. Why? Because this Codulegaster is simply waiting until an amphipod or a crustacea will come here and they will be eaten. Another group of species of dragonflies, they say, well, change its color is really definitely not my thing, and it's really boring to be buried all the time like this and waiting for food. I prefer to travel around, but I don't want to be detected. And what they do, they let grow like a lot of sediment, detritus, anything you want on them. This is Libellula depressa, and you can imagine when you find that exuviae, it will be dirty. So to see if it has dorsal spine, lateral spine, sometimes you really need to be working like a Picasso with a quast or something like that to clean all this with a pencil, to clean actually all this exuviae from the dirt. And some of them, like for example Ortetrum, I have some, I just can't remove the dirt because Ortetrum in Turkey, like Teniolatum, for example, they live in really muddy places. They are completely muddy, and once they come out of the water to emerge, they have to be hurry, because at one point the mud it's so warm outside that the mud that they cover them will start to cook and to bake like pottery, I will say. And sometimes they are killed before they can't emerge because the mud is getting too hard. And it's almost like you cannot clean them afterwards. But why do they have all this strategy of having, for example, changing color, of having buried themselves? I told you a bit about that. It's because they want to find food. Most, of course, being avoided by predator, like a fish or a kingfisher, for example, but mostly to come closer to their food, because these larvae are, I will say, a wonderful organ, and many persons say it's unique in the world. I will say in all this group. Well, it's not. I have to say my specialty is the zooplankton, and if you look to the rotifera, they have like a momentum to grab some prey and to eat it. But the rotiferas, they are even more wonderful. I have poor dragonflies because, well, actually they use this organ to grab a prey like this, but also at one moment when the prey is eaten, they will return inside their body the same organ to remove the skeleton of that. So it's like a multifunctional tool. Well, for larvae, this is still an amazing organ, and there is a lot of research on that. This is a typical one, as of course you remember, this long antennae, it's Scalopteryx. Which species here? Very difficult to tell because it's this one is Scalopteryx splendens. And this gape here, why is it here? I have no idea. This is the prementum, and these are the teeth of, this is attached to the head. But this, you can see here, and I will show you more picture, it's actually this movable part of the prementum. Now I will show you a small movie. I hope that internet will allow that due to the speed. And you have to watch carefully. I will remove this here to have the access to my mouse now. And if you look, let's have a look if it starts. No. I have to remove this laser pointer to switch it off. Yes, now I can start. Look carefully. It will go fast. Too fast. This is Scalopteryx splendens, and if you look in slow motion, it will grab a mosquito larvae and it will start eating. I will show you one more time in real speed. This is really, really fast. And actually, this is how they eat. Not easy to film, of course, because you almost need like a high-speed camera. And here, for example, I will show you another one, which is Anax imperator, and it's actually sometimes funny because dragonflies. Sorry, it didn't work. Yeah, because this is Anax, and you can see this mosquito larvae. And if I was them, look how fast it goes. if I was a mosquito larvae, I will not move, because dragonflies larvae, all of them are typical hunter due to movement. So you can see the one moving very quickly. These are the poor guy that they will be most probably being eaten like a spaghetti. And when you have, for example, anax, it was really interesting to see species like anax or Aeshna. I had once Aeshna serrata, and we had a kind of connection because it was eating, and every time I arrive to bring food, this larvae was afraid of me and starting to swim in the aquarium. But at one moment, it really definitely recognized me as you bring me food. And at the end, it was even waiting out of the water almost with the head to grab some food. And sometimes, look, this one was moving just a bit too much, and it's also eaten. And you will see, I will bring some food. It will be like a group of it. You will see the pipette tip with those poor mosquito larvae, and it will appear on the upper part of the video. And this, you can see that, and then poof, this, and they can eat a lot, really a lot. And you will see all this possibility on that. So how does it work? Well, I will grab back my laser pointer, and this is a prementum of Aeshna mixta, and in fact it has several parts. This is the major part called the prementum. This is the part underneath is the submentum, and these are the labial pulp and the movable hook. And this makes it actually like this organ of really of hunting organ. They have several shape. For example, the Aeshnidae have a flattened one. Well, for example, in Coduligaster, well, if you look to that, you really definitely don't want to be a mosquito larvae. Well, actually, I will say if you have to choose some species, don't be never a mosquito larvae, because everybody will eat you. And but this one is also a way to grab actually like a Gamarus, and it will eat it. You can see here Libellula, for example, with this two labial pulp open, and you can see the teeth here, for example. And if you still choose to be a mosquito larvae, well, if you have to die, and you will, try not to be eaten by Zygoptera, because it takes times. By Zygoptera, like an anax, you will die within a few seconds and you will be like a spaghetti. And but Zygoptera, it takes time. And for example, here, Sempecma paedis, this is Chalcolestes viridis. And by the way, you can see some species sneak around as well. These are vorticels, and these are kind of zooplankton, I will say. And well, they are pretty messy when they eat. And this time of thing grows on the exoskeleton of the exuvia, and they just profit actually from the mess. But if you look, for example, in Sempecma paedisca, the larvae is almost transparent, and which is interesting because you can see actually the mosquito is, of course, is here. You can see the mess they make. But if you look here, it goes obviously up to here, and then it's starting to be green. So poor mosquito is already eaten. Anax, for example, well, these are incredible hunter, because if you look to that one, for example, with Arcelus aquaticus, well, it just smashed actually cut the poor animal in two and eat. Here, for example, Aeshna uncea, having a good meal as well. Or, for example, on this picture, Aeshna mixta, you can see here the prementum, you can see a bit of the submentum, and if you look to this picture, you can see clearly the submentum part and the prementum and the poor mosquito. But in fact, there's a few things that we really still in research on this topic, because you can imagine, as I told about the world of health, for example, if you understand the mechanism and the way things works, then you can use it also, for example, like for human body. Imagine if you lose your arm, this looks pretty much like an arm. And also here, it is not like it's in science fiction, especially from universities in Germany specialized on this topic, like the university in Kiel. and a friend, Sebastian Busse, well, he works really on this approach of morphology of larvae and the biomechanics in of it for example, robotics. And it's he works with Professor Stanisław Gob, and this is what they make. For example, here, this is the morphology of the head of the larvae with this prementum. These are the labial pulp, and you have the prementum here and the submentum, and here clearly he calls here postmentum, submentum. There are different names, but it's the same. And this is how it works. And trying to understand this constriction, for example, you see here muscles and like the trigger muscles and the catapult muscles. Then they start, for example, like here, making 3D print for robotic using the same system for this. Well, I have to say there is a lot of work to do, and what they do is really wonderful. But for example, the problem is speed. Imagine if you have, how can I explain it? Because you cannot see me maybe clearly, but if you want to increase speed, you can use, for example, a movement of an arm. But if you extend your arm and you go back, you will have, let's say, at one moment like a maximum velocity, but your bones, your tendons, and your muscles will not allow you to work to have like a very, very quick movement. For that, for example, you can use like having a click system. Like, I don't know if you see it, but my thumbs, you can maybe hear it. Then I increase velocity because I just have a fast movement due to a click system. But here it is in the prementum of the larvae. It is not like this. So at the beginning there were suggestion that it might be an hydraulic system, but in fact we know now this is not. And now the new hypothesis is the combination of the catapult system of this muscle I showed you in the picture before. But the problem is it's still an hypothetic, and they are still working on it, because for this moment I will say, if somebody asks me how do they just grab, as you show in the short movie, so quickly a prey, well, we just don't know. And they do grab prey because, for example, here you have Aeshna grandis, and tinka, I don't know the English name of this fish. It can be much bigger, but this is a young one. And at the end, well, they eat almost like everything of it. There is a very few left. And they also can eat on a very strange way. This is a picture I will show you from a friend, Carlo Galliani from Italy. And it is a picture. Here you have the exuviae. This is a typical picture of Aeshna juncea with the exuviae freshly emerged. And here you have the adult, but this adult will die. And why? Because there is a sneaky larvae here from most probably the same species, definitely an Aeshnid. But this larvae just came out of the water and start to grow, actually start like a cannibalism, start to eat on the adult here. If you look closely, and you can see, for example, this now, it happened for quite a while, because you see these air bubbles here. This is the breathing system of this larvae. So these larvae are really, it's like, imagine for you, if it's your size, it's like you have like a huge hamburger to eat. This is, and they still eat quite a lot of it. This is, by the way, you see these larvae out of the water. They can survive. This is really amazing. They really can survive for a long time out of the water. Sometimes legs go dry, for example. Of course, it's a very long time. It's definitely a problem. I have seen, unfortunately, I've seen that with Codulegaster larvae. I wanted to be sure of which species it was, so I brought that home and tried to bring them out up to a Codulegaster adult to be sure, especially when you work on photograph or funky, you want to have the right species. And one Codulegaster, it was at the time Heros pelionensis, just come out of the aquarium I have, and I lost it at home. So I'm afraid I searched for it, I couldn't find it. And after a week, when I was cleaning the living room, I found the poor larvae completely dry, full of dust. I definitely had to clean better. But the thing was, when I decided to keep this dry larvae and I put it under water just to remove all this dust, and I realized that one leg was moving after a week in the living room. So this is pretty dry, I will say. And what did happen? I put it back in the aquarium when I saw maybe it could be still alive. But the fact was, it was floating. It couldn't. Larvae are swimming underwater. They live underwater because their density is higher than the water, because they have a lot of water in their body as well. But this one lost a huge of the internal humidity, I will say, and was floating on the water. So I built underwater a bridge with stone, put it on it, and after 2 days, didn't move for 2 days, and after a few days, it was back the worst nightmare for all those mosquito larvae I put in the water. Are they the boss on the water? No. As I told you, a fish can eat larvae, or for example, a kingfisher here. This is a kingfisher with Cordyligaster, and actually sometimes it's possible to identify also this Cordyligaster. For example, this is a kingfisher pellet, and if you look here, you have this is the typical prementum of Cordyligaster, and I know that this picture was of these pellets come from Switzerland, and there are two species there, one without lateral spines and one with lateral spines, and here you see clearly lateral spines, and all the larvae, whether you are Zygoptera or Anisoptera, if you are a female, you have on segment 9, which is here, this is 10, this is 9, they have an ovipositor. This one, you don't see one here. So it was actually a male of Cordyligaster boltoni. People ask from time to time, for example, when I travel and I make those pictures, how do you make those pictures underwater, for example? Well, it's not that complicated, but for example, this is a wonderful little house in Sardinia we have rent, and when you come in the living room, for example, and there's a guy showing us around for this is the place to sleep and this is the place to eat and so on. Well, the living room, it was really wonderful, and we planned to stay for a week there, and say to the guy don't come for 1 week, we will clean the whole thing, because actually when the guy left after 1 hour, the whole room was looking like this. And here you can see a tripod with a camera and a laptop, and this is how I make those pictures. And of course, I stop a few beers, but I stop because otherwise it will be later on sharp. But in the field, for example, here you can see me, for example, making those pictures, and I use actually like free flash. And this one, I put a plastic bag, because once it was swimming also with these Cordyligaster larvae, but it survived somehow. So, but it is all more. You have one camera with an infrared systems, and I put on the background of this, on the aquarium, I put like a greenish background or bluish background. I try several, but I would say blue look really like artificial underwater. And so it was not nice. But the most important, if you try this system, use sand also at the bottom, because otherwise you will have a lot of reflection, and it will be actually way more. No, you will not have a good light at all. And why do I make these pictures? For example, well, it's to make presentation, to write books, because I think working on dragonfly larvae, because it's not known that much, well, it's a very useful tool. You can also, for example, especially when you're a specialist and there are groups like odontology groups from each country, you can make, for example, like excursions, sometimes for example, with children, and it's very nice to catch dragonflies and to see them like in an aquarium, and see them within a few weeks to see them emerge. This is Ashna isosceles, and they were I had like 3 or 4 in the aquarium, and they were all emerging at the same time. And with excursions, well, it's very nice because you can share a lot of knowledge, and sometimes, for example, you can, especially in rare species of dragonflies, you can spend hours searching the adults while searching the larvae. If you are at the right time, well, you can have rather a lot. For this presentation, I would like to thank, first of all, Ewout van der Ploeg. Ewout is a very good friend, and we made a lot of travel together. This is not Photoshop, by the way. This is Leucorrhina rubicunda, and this guy just attract them. How does it work? If you look carefully, this is the moment Leucorrhina rubicunda emerge in the season, so it will be somewhere like April or May or something like that. But this is the shadow part of Ewout, and this is the sunny part of Ewout. And these species, sometimes they emerge so much that you can have millions of them, and they will try to catch, even if for example, it's late in the afternoon, they will try to catch the last sunlight. And there are so many flying around that they will land on you, and they will definitely choose for the spot to have the most of the sun yet. These are other friends I would like to thank, because sometimes they help me with places or with pictures. This is an association from Holland, which is really fantastic, because you can present a project to them, and especially when it comes to entomology, and they can provide you money to make a trip to Turkey or to Greece or something like that. This is a company I work with and the publisher. And I would like to thank you all as well, and also enjoy and go in the field and try to study as much the world of dragonfly larvae. I thank you very much. I will stop sharing my screen.
Okay, thank you for your lecture. And does anybody has any question?
I would like to have one question, if I can, regarding the topic of the. You said at the beginning that when dragonfly larvae are in an environment where there is not much food,
yes,
they in fact grow larger than in environment where they have a lot of food.
Yeah.
And I wanted to ask if that is also reflected, like if there is a variability in the size of the adults, because like what you showed in the picture, like the difference in size was really great. So
in adult, I think once they have emerged, I don't know exactly about the let's say species within the same species, the variation of the adult. I don't know. Let's say from experience, I don't have the feeling that they are really. It's not like I have a small Eschna grandis and a long Eschna grandis. I think they almost reach almost the same size. There should be a tiny variation, I guess, but not like in exuviae or in larvae, for example. And so I'm not sure about that. I don't think so. I think on the other hand, maybe on Corduligaster it works to see those differences because they stay a long time underwater. For species like Lestesdrias, for example, they have a smaller variation as in size as an larvae. Then it's because they stay too short underwater, maybe to see those kind of differences. But also I have to say this is it's more like I have seen that. It's clear that much more research should be made, for example, to study the size of the larvae the moment they molt, and also the abundance of food around. And for example, some people did that for like Robert. This adult book of dragonfly I was speaking at the beginning of the presentation, well, at the end he has all kind of measurements of larvae. Like, for example, from the egg, he started to measure some of them. This is a huge amount of work. I got, for example, also in size or in variation. For example, I got also questions, especially for people who work with larvae instead of exuviae. The reason why I study exuviae, I told you, because I don't have to kill a lot of animals like this. I just have to take the empty shells. And some exuviae, they can, I guess it was a museum in Switzerland of Neuchâtel or something like that. There are exuviae which were very old, and you can have, you can check, for example, these exuviae, and after 50 years or 60 years, if they are well preserved, they are still looking the same. The problem with larvae is also that, first of all, you have to kill some, but you have all kind of stage and some criteria, like in Corduligaster, I was talking about lateral spines. At which moment does it appear? I just don't know, because I don't know that. So there is definitely a lot of work to do about those measurements for food, and the relation with the size of the larvae and the exuvia.
And just to clarify one more thing about this topic, the hypothesis why they grow larger in the presence of less food is that they need like a large amount of energy at one time for the final molting, and like if they just have gradual energy supply, then they can never get like that critical mass of energy to do the final molting. Or
it's if I understand you well, it is that they have to wait. I think the most critical moment in the life of a dragonfly is the moment they go from the larval stage to the adult stage, and I think they need this time of. I think they can like feel of like they have an organ or a sensor, I don't know, that says, okay, now I have enough to molt, because sometimes I have seen fully grown larvae, especially when it comes to Leucorrhenia pectoralis, for example. I've seen that several times in that species, and also in Cordulia. A larvae that come out of the water maybe try to say, okay, I will emerge. I don't know what, of course, difficult to say what they are thinking, but at one point, it will not go up to a branch to emerge, it go back in the water. And this type of larvae, for example, you will find, for example, like now, if you look in the ditches, you will find like fully grown larvae almost close to emerge. But they somehow decided to say, okay, I will stay one winter more underwater, and I will come up the year later. Well, if you look to the shape and their body mass or something like that, well, they pretty much look the same as the one that emerged already the same year. But somehow they can maybe, I think the answer is because they see I just maybe not have enough energy to make this transformation from the larvae to an adult.
Okay, thank you very much.
You're welcome.
I have question. How is possible that larva of anax is much more bigger than larva of eschna, but adults, for example, anax imperator and eschna cyana has nearly same size?
Yeah. If I understand you well, why, for example, the larvae is almost as long as the adult, for example, is that what you say?
That the difference in size of larva between anax and eschna is much more higher than difference in size between adults.
I don't know. Maybe food, but I don't know. Maybe the amount of molt they do, because Robert, I never made some study on that, but I know that one species, for example, can vary between 10 molt up to 15 molt. So I think food is the key on that. The way they say, okay, within 10 molt, I have already enough energy, so I will not have to grow further. I think this is the reason of this variation.
Okay, and another question: how many species in Central Europe and in Southern Europe is able to perform 2 or more generations per 1 year?
It's important to say, like as you said, like in Central Europe, because, for example, if you go to Spain, one species will have let's say one adult stage in spring and one other one in the summer and one other one later in the in autumn. It depends how many species. I will say almost will look more or less to families. And for example, definitely in Central Europe, I will say Eschnids. Most of them, not all of them, like Anax ephipiger, it can go rather quickly within the same year. But like other species, and especially if you go to mountains, like cold system species, they will definitely have more years. And even a Zygoptera that's in Czech Republic, rather low, for example, in which species can I say? Maybe Ischnura elegans, for instance. Well, it could be that it will be within the same year, it will be an egg, an larva, and an adult, rather that if it's more higher, for example, in systems, then it will take one winter as a larvae. So there is a lot of variation within the species. Over Aschnitz, for example, like Aschna affinis, which is definitely a species that lack, well, let's say warm system. It will take maybe I don't think 1 year, I think 2 years. But like Aschna juncea will definitely stay 2 to 3 years underwater. So I don't know exactly how many species does this strategy, because within the same species there is the possibility that there is variation as well.
Okay, and which species are able in Southern Europe perform more generations per year?
In Southern Europe, well, rather a lot of Zygoptera can do that. One of the typical is Ischnura graelsi, which is in warm system. I have seen that also, for example, if you go to even more south, like in Canaries Island, like Tenerife or Gran Canaria, for example, I have seen there, it was Tenerife and Fuerteventura. On Tenerife, you have Ischnura senegalensis, and on Fuerteventura, Ischnura saharaensis. And I was there in February, and there were adult emerging. And so once you are reaching the south, they can go on all year like this. So they will be flying like maybe not as a larvae when it's completely a dry season. You can see that when I was talking about Sempetrum depreciusculum, that sometimes the system is completely dry. You have that clearly for species like Lestes macrostigma, which is typical species from the early spring. For example, I was on Cyprus. I didn't saw any Lestes macrostigma at all in May because they were all dead already. But I could find completely dry systems of lakes, and they were all waiting as egg the season of the season actually to well, I don't know exactly, maybe autumn, something like that, to come out. So Zygoptera does clearly that, and Isoptera, as I said, Anax ephipigia is typical one, which is really fast growing. It does, I don't know exactly, but within a few months already, it goes from egg to adult. And you see that clearly for these species, for example, when they fly from the south and they start laying eggs in May or June or something like that, and later in the season you can find them back emerging in later. So it's definitely possible. Or when they arrive in late September, for example, which is the most common one, and you have to wait just for one winter, and they will emerge rather quickly, and then they fly away. So there are also strange behavior. For example, as larvae, as I said about species from that use another strategy, also as an adult, Sampecma. Why, I think, do they use this idea of staying as an adult during the winter and going laying egg in the spring? Well, I will almost say it's a clever choice, because when they stay as an adult during the winter, they will go and lay eggs at the moment that most of the larvae are coming out of the water, which means that in the period of June, you have a lot of dragonfly flying around, but a few larvae underwater. So Sampecma decide to do the way around. Maybe, I don't know, it's an hypothesis, but I think it could be possible that they decide to do it like this, because at that moment, they can say the moment the larvae is underwater is the moment with the less competition. But it's an ecological trade-off, because on one point they can say, okay, it's cool, there are not a lot of larvae, this is the moment I will be underwater. But on the other hand, for example, like 2 weeks ago in Holland, it was really cold. And then they have also to survive this harsh condition of weather, for example.
Okay, for Simpec mind, also Lestes, I think that is important that they are common in habitats with in periodic tools, and that is avoiding of the drying in the winter or in the autumn.
Yep.
That, as I found habitats which are dominated by these genera, it's mainly periodic stools.
Yeah, it depends the species. When you have Sempecma fusca can be completely dry but for example like Sempecma paedisca, we have in Holland both species. Paedisca is really having a hard time now, especially with the competition with Sempecma fusca, because Sempecma fusca will win at one point. I think Sempecma paedisca will disappear, but in paedisca they stay. The water system doesn't. They stay full in water, but fusca can definitely have this periodic system of temporary water. Are there more questions? Okay. Well, I would like to thank you because it was interesting for me. I have to say, I give quite commonly a presentation. Working on a computer, it's seeing a screen of instead of seeing a public. Well, I have to adapt on that. But on the other hand, the nice thing is, of course, that you can. I can do, for example, a presentation. And Saturday I was giving one, as I told, of Svalbard, for example. So it's very interesting that I can reach a lot of people like this. And I hope that you will, in the future, of course, study the adult of dragonfly. They are amazing and really interesting. But spend time and study their larval time, and especially also during your study. For example, I have already in Holland several master students. I'm working sometimes with university, and who spend, for example, their master working on dragonfly larval systems. I will definitely advise you, for example, maybe it's really possible in Czech Republic. In Holland, unfortunately, there are very few professors that says, oh, dragonfly study is a very nice study, topic of research. And for example, Germany, they have really really nice friend who are professor there. And if you definitely want to work further on dragonflies and you don't have the possibility to do that in Czech Republic, don't hesitate to contact me. I can bring you to friends who work from university, and you can really have some nice projects.